Hello everyone and welcome to our CENTO webinar, When Knowledge Turns into Value, Variant Reclassification. In today's webinar, we have with us Gabriela Elena Oprea, Senior Vice President of Digital Products at CENTOGENE. During the webinar, she will discuss the following topics: variant classification and reclassification at CENTOGENE, case studies based on internal knowledge at CENTOGENE, and the importance of structured communication when variant reclassification occurs. If you have any questions, please enter them into the Q&A chat and we will refer back to them at the end of the presentation. It promises to be a very interesting webinar, so let us begin.
Thank you, Lina, for your introduction. I would like as well to thank you all for joining us today. At the beginning, I will focus on the variant classification process at CENTOGENE and then give you insights about variant reclassification. Next, I will like to proceed with case studies and to end with the importance of structured communication when variant reclassification occurs. So, let's get started with the variant classification process at CENTOGENE. CENTOGENE continuously identifies new genetic variants, and we have implemented the ACMG guidelines in our variant classification workflow. Variants detected in Mendelian disorders are classified based on the likelihood to predispose or to cause the observed phenotype into one of the following 5 groups: pathogenic, likely pathogenic, uncertain, likely neutral, or neutral. During this process, we evaluate all available information we can gather from external sources and compare it against internal observations. On this slide, we have depicted the most important criteria evaluated during variant classification process. This data is gathered and analyzed at genetic, clinical, and, if the case, biochemical levels. Information coming from general populations and diverse subpopulations, especially from the internal observations, are combined and then used for applying ACMG guidelines. All evidences are then subjected to an independent curation, following systematic and standardized workflows, updating them quarterly, and provide fully transparent evidences. Thus, we reduce the risk of misclassifying the variants. We have observed that more diverse the population is, higher the tendency to classify variants previously reported as pathogenic or uncertain towards benign. A previously rare variant, usually on Caucasian-based observation, becomes normal polymorphism when initially underrepresented population were taken into consideration. And it's really important to say that currently we have internally patients originating from more than 115 different countries worldwide. Concerning the external sources, we are using during variant classification, as I mentioned, different external databases. I have depicted here, I will say, the most 3 used external databases. It's the HGMD of the Human Gene Mutation Database, is ClinVar and NOMED. And there are already a lot of publications where some limitations and challenges are openly addressed. There is so far, and we have to be honest, no unique source to be used when classifying variants. That, for example, 25% of the disease-causing mutations in HGMD are most likely to be misclassified when looking at autosomal recessive disorders, or that, for example, 17% of the clinical variants are discordant classified by submitters is already known. So the issues we have identified are about the evidence transparency, it's about curation of the data, it's about the missing clinical information on the patients, about the cohort diversity, and at the end, no dynamic communication. At the end, these limitations will impact the patient at different levels, the level of diagnosis, of the future planning for the families, will impact treatment options, and in the worst case, the patient can't be misdiagnosed. So, to overcome these limitations, these challenges, CENTOGENE has developed and implemented its own knowledge-based database. The variants which we select to be reported are first analyzed against the clinical information, against family observations, and during the reporting, the clinical assessment and genetic diagnosis are summarized, and these will feed then back the CENTOGENE database, which we call CENTOMD or CENTOGENE mutation database. And here within this system, we compare the own gathered data against the selected external sources. Currently, and this is I think very important to mention today, is that 58% of the variants we have reported are uniquely described in our database only. You don't find them in any other database, and the value we bring for all these high portion of variants, which we call them clinically relevant, is that we associate them with clinical information and we link them to human rare disorders. The data coming to the database, they are not based only on publications, they are based on the real diagnosed cases, and behind every case stays an issued medical report. So now let's get on the next level, namely the variant reclassification. So once a variant is classified, no matter the class, every new observation is monitored and then used for variant reassessment. We at CENTOGENE allocate substantial resources to review clinical significance for the detected genetic variants as new evidences become available. We have a dedicated team which is focused only on the monitoring and further improving the process, as well to bring automation within this process, and the team acts independently from the reporting team. We call it the second independent knowledge review, but the team interacts closely with reporting team in approval processes. If there is insufficient information to reclassify the variant, CENTOGENE will continue to monitor and evaluate if the case additional information as they become available. But if there is sufficient information to reclassify the variant, CENTOGENE initiates the variant reclassification process. How fast we react depends on the change. If we have a high-risk change, meaning there is a change which impacts the diagnosis, we react immediately. Immediately means within 24-48 hours. If we do have a low-risk change, so the diagnosis is not changed, it could be a likely pathogenic to pathogenic classification. Then we took as maximum 1 week to issue these reclassification reports to the corresponding physicians. The system we have developed evaluates and notifies the expert team when, for example, the frequency criteria of variant changes. We call them warnings. They are received based on either external sources, but more frequently they are coming from internal observations, since every new family, every new case was going to feed the curation database. And they are going to contribute to the calculation of the internal allele frequencies. Zygosity is one important factor. For example, when the variant is localized in a gene and the gene is linked to an autosomal dominant disease, and we identify a homozygous patient, then the system will automatically build such a warning, and the expert will pick it for manual resolving. We as well very carefully review and take into consideration information related to functional studies, to disease mechanism, or how, for example, the DNA change will impact the protein domain. So we go to the literature, we check carefully and document this information in order to share with our expert team. But I have really to admit that the team gets really excited when new observations are made based on a new case, a new family, or new cohort analysis. It could be that a previous segregation observation is confirmed by an independent family, or it could happen that we observed new co-occurrences. It could be that new genotype to phenotype correlations are observed, and so on. So all these are coming to the database, and we look based on the new observed evidences if can be any jump between the 5 classes we have established at CENTOGENE, and then we can end up with refining frequency criteria or specific criteria on the gene, disease, and case level. And I'm really as well happy to know that this dedicated team is really enthusiastic and ready to react whenever such a change is detected. Currently, we have, I will say, many patients analyzed for rare disorders. Recently, in fact, we reached the 200,000 individuals in our database, and we confirm more than 3,400 associated phenotypes in the context of rare disorders. Keeping in mind that these patients are originating from more than 115 different countries worldwide, we own a unique patient cohort. The 6.7 unique variants or DNA changes we detected, we keep them associated with clinical information, with family history or other medical evidences. For the clinical information, we have implemented the HPO nomenclature in order to standardize such information and be able to easily calculate similarities between different patients. So once variant is reclassified and approved by medical experts, then we proceed to identify all the patients where the variant was reported. We issue for all of them reclassification reports to the corresponding physicians. In this manner, we bring our current understanding to our partners in a proactive manner. The most of the variants were uncertain and reclassified based on new evidences, either on higher or lower level. An added value to our classification and reclassification process is represented by in-house established biomarker and biochemical testing. Using a biomarker either as diagnosis tool for lysosomal storage disorders or in regular monitoring of the disease progression can be of high clinical value for the patients. The biomarkers are used to elucidate the molecular pathogenesis of the disease, and CENTOGENE allocates substantial attention and resources to even develop new assays, new biomarkers that can improve the condition and the prospects of the patients affected by lysosomal storage disorders. We have analyzed, and here I have 7 selected genes for this analysis, where variants described and reported externally were compared against the internal database. We took in this comparison the 2 most used external databases, ClinVar and HGMD. As you know, in ClinVar, there are different submitters. Every submitter will add their own classification, HGMD as well, a curated database, and every variant is linked at the end with a unique variant clinical significance. And it was really interesting as well for us to see that 8% of the variants we commonly detect in ClinVar and internally are discrepant classified. But when looking to HGMD, this percent of discrepant classifications reach 10%. We can translate, of course, these numbers into the risk of misdiagnosing the patient. And the 10 or 10% misdiagnosis rate is really, really high. So now I would like to proceed to introduce to you 4 such examples where CENTOGENE reclassified variants based on our continuously analysis of internal observation. My first selected example is a variant localized in the GBA gene known to cause Gaucher disease. Gaucher disease is a lysosomal storage disease, rare, inherited in an autosomal recessive manner. This variant is located at position 1574. It's a substitution of G2A, which leads at protein level to an amino acid exchange, which we call missense, where the glycine at this codon position 525 is replaced by an aspartic acid. The variant is not described elsewhere, and we still knew it based on the external as well internal observation. These kind of mutations of the missense variants in GBA gene have been already described as pathogenic. In fact, 96.5% of these variants have been reported to cause Gaucher disorder. The variant is rare, is not described in NOMAD, and predicted to be benign by different prediction software. Concerning the conservation, it is weakly conserved as well the nucleotide as well the amino acid position. Initially, we detected this variant into 2 heterozygous individuals with no clinical information provided by the physician, and the biomarker levels were within the normal range. So initially, we classified this variant as being uncertain. Well, in the meanwhile, we identified at CENTOGENE 2 independent individuals, one being homozygous for this mutation. And the clinical picture was as well suggestive for Gaucher disease. The final biological evidence which was used to reclassify the variant as pathogenic was coming from the biomarker analysis. We checked the enzymatic levels, and the activity was within pathological range. Sorry, and the biomarker levels were found to be within the pathological range. You see here the reference of this biomarker. The internal observed frequency was as well rare, so we reclassified as pathogenic. We had our second independent individual with 2 heterozygous GBA variants. We call them most likely in trans, and we do that as well based on our very specific biomarker used for Gaucher, lyso-Gb1, since it was as well pathological. And we know as well from our own data that the carriers of GBA pathogenic mutation, no matter if they are just one or they are in cis, so 2 GBA mutations, but in cis, the biomarker levels will stay within the normal range. So based on these 2 observations, we have reclassified as pathogenic. My second example is an intronic DNA change, which leads to a substitution at position +6 in the intron 2 of IDUA gene. It's a substitution of C to and IDUA gene is known to cause mucopolysaccharidosis type 1 disease, which is as well rare lysosomal storage disease and inherited in an autosomal recessive manner. Initially, the variant was published as disease-causing mutation by HGMD. It was then reported as benign or likely benign in ClinVar, but no evidence or no explanation why. The conservation weakly conserved nucleotide but moderately conserved amino acid. From the prediction software we used was coming an unlikely aberrant effect on splicing. And from NOMAD was coming the information that 19 homozygous individuals are detected with a frequency slightly higher than 1%. Not to forget that NOMAD was released last year, and we detected for the first time this variant, in fact, in 2014. So at that moment, when we classify the variant as uncertain, this important information from NOMAD was not available. Anyway, in the meanwhile, we of course continue to screen IDUA patients, and really interesting, we had 5 patients in all 5 patients with clear indication that MPS is the suspected disorder. And for 2 patients, we have confirmed the presence of MPS 1 by other IDUA pathogenic variants. In 3 of the patients, we had clear pathogenic variants in other MPS-related genes. The internal frequency we have at the moment for this variant is higher than NOM-AD. We have 1.7%. And additionally, when we look at the non-MPS population, we have identified this variant in a heterozygous state in more than 700 individuals, and 90 of them being homozygous. And they were, carrying different clinical picture, and they were somehow age match controls. Very important is that in the same time, we have identified 4 healthy adults. All of them were homozygotes, and all of them, in fact, were parents of children with MPS suspicion. So at the end, based on all these cumulative observations, we reclassified the variant as being likely neutral. My third example is a reclassification of a variant, and I find it really, really interesting because it's I will say, maybe it's one of the favorite example I have, since it's coming from a common effort from CENTOGENE side as well from physician side to clarify the clinical significance. Initially, so we identified in a trio, and the trio was made from an index child with suspected diagnosis of Bardet-Biedl syndrome. The parents were unaffected, and we have analyzed all the BBS related genes we had in the panel. No clear pathogenic variant was identified, and the only candidate we had, it was this variant, intronic variant, which is located at +5 position. The prediction tool provided a hint that splicing may be impacted. It is a highly conserved donor splice of the exon 3, not reported. It was a very, very rare variant in NOM-AD, but no homozygote. Only one heterozygote was identified in this database, and in NOM-AD genome, no information about this variant. So we had the genotype to phenotype correlation. The index was homozygote, parents heterozygote, but we couldn't have at that time, any supporting evidence for pathogenicity. Nothing against, but nothing more. So we have contacted the physician, and we ask for either more information or to send us, if possible, additional relatives. The physician really open, understood very easily the situation, so he came back and told us that the family is consanguineous and is originating from Middle East. What we did first, it was to really check this Middle East population, and we couldn't find this variant in any screened patient which was not belonging to this family, even if we screened 36,000 such individuals coming from Middle East. So the internal frequency really low. And in the meanwhile, we received 2 additional family members. It was one sibling being affected, similar like the index, and another healthy sibling. Um, and of course, we analyze all of them together, and we observed a nice genotype-phenotype correlation. Both affected siblings were homozygotes, and the 3 healthy individuals were heterozygous for the variant. Knowing as well that the family is consanguineous, we reclassify the variant as likely pathogenic. But we have to admit that this would not have been possible without the implication and motivation of the physician to clarify the diagnosis within this family. That's why I would like as well to thank now to him for the support he had, and as well for all the information he shared with us. My next example, my last example on this topic is a missense identified in this gene SHH, Sawing hedgehog gene, which we identified a codon 290. There is a missense. Again, a glycine is replaced by an aspartic acid. And initially, we identified this variant in a patient, which was referred to us with the clinical suspicion of holoprosencephaly. Variant was reported in HGMD as disease causing, ClinVar reported as inconsistent, pathogenic, VUS, and benign. Predictions were inconsistent, conservation not really supportive. Then from NOMAD, we saw that 9 homozygotes were described, and this was for us a very important information, since we knew that the disease is an autosomal dominant disorder, and we will not expect to have homozygotes, especially unaffected. So in the meanwhile, we received another interesting family. We identified this variant in an homozygote state in an unaffected mother. The mother had a child with clinical suspicion of holoprosencephaly, and the child was found to be heterozygote. In that moment, we initiated the reclassification based on no genotype-phenotype correlation. And we did as well internal populational or cohort check, and we identified 52 individuals carrying this variant in heterozygous state. And it was no G2P correlation, since the most of the patients were healthy or asymptomatic and adults. At the end, we agreed with the medical expert team to classify it as neutral. So these were my 4 examples I wanted to introduce to you concerning the variant reclassification. And now I would like to say something about the importance of such a transparent communication when variant reclassification occurs. Well, we do our best to keep you up to date with the most accurate, clear, and transparent genetic information, and we have to do that because genetic knowledge constantly grows with new insights in disease gene associations and related pathogenic variants being daily detected. This makes, of course, variant classification, reclassification ongoing process that consequently can have a lifelong impact on patient care and disease management. In order to reduce the risk and harness the benefit of this rapidly increasing knowledge, we have designed a highly robust and dynamic variant reclassification program that enables you to provide you with the latest and highest quality data for the most up-to-date diagnosis. Four advantages of our variant reclassification program I summarize here. There is about proactive notification of every patient affected by the reclassification, the basis which we have, which is a unique combination of patient data from genetics, biochemical, and clinical. The variant data derives from a large and diverse and unique cohort of patients, and all the data we use in order to support reclassification go through stringent data curation and validation process. Here I have such an reclassification report example we have issued to our partners. Here is a reclassification from likely pathogenic to pathogenic. And it's really nice to see how external and internal observation are used in order to definitely confirm the presence of a genetic disorder. In this situation, it was the glycogen storage disease type 3. But what motivates us to be committed to invest effort in regular variant review is in fact the benefit to both physician and to the patient. We strongly believe that physician saves time and is relieved from doing him or herself this search. Physicians stay thus up to date with the most relevant genetic information about variants and disease. Physicians can provide a better patient care due to free of charge reclassification reports, and at the end is able to manage and treat the patients and the families in appropriate way. On the other hand, the patient has the right to receive definite diagnosis and must know when and especially why uncertainty is removed. If the patient is correctly diagnosed, his or her quality of life is improved due to targeted disease management. So, to conclude, variant reclassification is dynamic, is ongoing, an important process of CENTOGENE. We take the responsibility to do our best with the current knowledge and share it once becomes available, and acting in this manner we turn the knowledge into value, and indeed every patient counts. And here I would like to end my presentation, and thank you very much for your attention. Ina?
Yeah, thank you, Gabriela, for sharing your knowledge with us. Let us now move on to the question and answer part of the webinar. I see that we have received several questions from our participants. And the first one is from Elizabeth, and she asks, which information from request form is used for variant classification?
Okay, so at the end, all the information from the request form is used in some way. For us, of course, we are really very careful to see if we received information regarding clinical picture about diagnosis or suspected diagnosis from physician side, if there is any family history or is the only or the first case in that family, everything helps. At the same time, we look to see if any other previous test or analysis has been performed, if it was negative or uncertain result, and if we receive medical records like perform biochemical tests or blood tests, we as well capture and use when we do our variant classification.
Mhm. Thank you, Gabriela. I see we have another question from Eric, and it goes like this. As a clinician, once I receive a report with a VUS variant, what can I do further to clarify the significance of the respective variant?
Okay, so I think this is a very good question, since indeed it should be a common effort to clarify such difficult situations and uncertain variants, and at the end really frustrated, I think, for the patient. As physician, I think what you can do is to provide complete information as well as specific information about clinical status, about the family. But in the same time, after we may issue one report, to provide us, if there is a case like follow-up studies, it could be that based on the uncertain variants, some biochemical tests were done, or some other family members were tested. So if we will receive this kind of information, we can reevaluate based on the current knowledge, and then coming with an improved variant classification to the patient.
Okay, thank you. Yeah, I see one more question which is sent in by Mirielle, and the question goes like this. When reclassifying a variant, how often do you search for additional information on a variant?
So I will say like that. So we go to search for additional information on the moment when we report the variant. So we go and have a look if all the data we have in-house is up to date. We can do it as well on the request. It happened, yeah, that some additional information was received, and then we do it again. And at the same time, we do it if it's not these two cases, we do it regularly. So every new update, every new case leads to an automated rule application within the system, and this is done almost every day.
Okay, thank you. I see another question, and it goes like this. Does CENTOGENE perform functional analysis on request?
Unfortunately, not yet. So we do not have, but we do collaborate, and you have to send us if you have such a request. Is we collaborate with the University of Rostock, and there we have a lot of enthusiastic researchers, and we do collaborate with them for some projects. So if you have such a request, I would say send this request to us, and then we get in touch with our collaborators from university, and then we can come back to you with an answer.
Thank you. Yeah, we have another question. So it goes like this: how often is the variant classification carried out, and in approximately how much times, and so how often is CENTOMD updated approximately?
Okay, so I will say we do reclassification once data is available. New data is available. This can take from weeks to years. The database is quarterly updated, but of course we, as I mentioned, we enter all the data as analyzed and processed, and have this kind of warning system that the system will tell us, here I detect something, you may have a look. This will end up with a manual solving of the warning, and then we try to do it in a quarterly base. But as I said, if we do have any high risk warning, we react immediately. But of course, we are always depending on availability of new additional information we can gather.
Okay, thank you. Yeah, I see another question from Michelle. It goes like this: how do you proceed with genes with the PSO genes? Like for instance, GBA has 96% homology.
G with pseudogene.
Yeah, with GBAP1, do you review if the data available for variant classification differed between both genes?
Um, it's a very good question. So when we establish and validate the test in-house, yes, we do take into consideration all those genes. If it's GBA or PKD, we really took into consideration the pseudogene in order to make sure we do not call I will say the variants of the pseudogene. This happened in the past when the technology or the technique was not so well, I will say, evolved. Nowadays, there are a lot of ways, and the bioinformaticians we have in-house to exclude such, most likely, we call them artifacts because they are coming from pseudogenes. And I have to admit that some of the variants, which in fact, for example, for some genes are in some other databases, are still in fact detected on pseudogenes. So we do our best during establishment. We validate the variants we detect, like real positive, real negative, and as well the false positive, false negative. And once the validation is approved by the expert team, then we go to production. So meaning in the production diagnostic is only use an validated test.
Thank you, Gabriela.
Yeah, thank you as well for all these interesting questions and for your attention.
Yeah, thank you, Gabriela, one more time for sharing your insights, and thanks to all the participants for joining our CENTO webinar. If you have additional questions to Gabriela, please don't hesitate to send them to her directly, and she will be happy to answer them for you.